Hello and welcome, everyone. This is a side session at the UNC Water and Health Conference. Thank you for joining us. I see we have almost 100 people on, and we're going to go ahead and get started. I want to say, first of all, this session is sponsored by Chemonics. So thanks to Chemonics for sponsoring the session. I think we've got a great lineup for you today as we explore the state of the science for SARS-CoV-2 monitoring and wastewater. This is an area where a lot of us are working, and indeed there are many others, great groups around the world that are leading research in this area. And it's an exciting place to work. We're learning a lot, and it's actually for me at least, it's been a really encouraging example of team science and collaboration across institutions and countries. So thanks very much, first of all, to all the speakers, but also thanks to everyone for joining and for UNC for hosting. Just want to say that the format for today is we'll have Professor Kyle Bibby from Notre Dame give a keynote presentation, about 20 minutes. Then we're going to have lightning talks, which is a format that was used for another recent SARS-CoV-2 wastewater surveillance conference that I really loved. Really punchy, small talks where people can share highlights from their own research. And then we'll have a moderated discussion. So what I would ask from all participants is that you please put your questions as they come up in the chat function of the Pathable website, not the Zoom. The Zoom is disabled for chat, I think, but the Pathable site will be taking your questions. You can put your questions there, and we'll be collating them, recording them, and directing those questions to specific speakers. Okay, I don't have anything else to say. This is really exciting. Thank you so much for joining. I want to pass now over to Kyle, who will introduce himself and start our keynote for this session. Thanks.
Great, thank you, Joe, for the introduction and for putting this together. My name's Kyle Bibby. I'm an associate professor in environmental engineer at the University of Notre Dame. And my talk today really it's my perspective and an overview of the state of science. Of course, there's a lot more going on that I could possibly cover in 20 minutes, but I kind of want to set the stage for the rest of our presentations and discussion, as opposed to focus on one specific research project that we have going in our group. So I'm sure that many of you here are familiar with the fact that the RNA of the SARS coronavirus 2, or the causative agent of COVID-19, in addition to being detected in nasal swabs and saliva, is also detected in feces in the majority of infected individuals. The data sets describing this are actually probably not quite as good as we would like, but you can see here a shedding chart put together by a postdoc in my group, Aaron Bivens. You can see the wide range in shedding detected of infected individuals in stool. And so that gives rise to, I think, two important research areas that I'm going to talk about today. So one is that there's been at least one credible report of infectious virus detected in stool. Most studies that have looked so far have not found infectious virus in stool. And there appeared to be some conversation today during one of the plenary sessions of infectious virus potentially being found in wastewater. Secondary, where I'll spend most of my time, is this idea of using this viral RNA that's found to monitor for cases within a specific community or sewer shed. It is important to note, and I'll note this multiple times, that in fact the detection of the RNA does not directly correlate to detection of infectious virus, right? And there's lots of different mechanisms by which those two are separate measures. So the first topic is this idea that perhaps there may be infectious virus potentially present in wastewater, or at least that this is something that we would want to be aware of. So our group, in collaboration with NIH Rocky Mountain Labs, just conducted a study where we spiked in culturable SARS-CoV-2 into at different concentrations into wastewater to estimate overall persistence. As you'll see, that this data is a little bit noisy because we're only able to run a limited number of tests, but the persistence is relatively low with a T90 or time for 90% inactivation of about 1.7 to 1.9 days, which is much lower than we'd expect for other viruses. And you see it's very efficiently inactivated by heat treatment here as well. So efforts to pasteurize wastewater samples before processing, for example, appear to be highly effective. You'll see we describe in this paper measurement of RNA as well, and we see very minimal decreases in RNA in these same experiments. And so this really highlights the fact that RNA is not equal to viable virus. We have ongoing efforts looking at disinfection of SARS-CoV-2 in water and wastewater matrices using a couple of different approaches. But the available data that we have from surrogate studies and data that's emerging on SARS-CoV-2 really suggest that this virus is not going to, say, reset our water management strategies. It's not an especially tough environmental virus, and while it may be detected in wastewater, it doesn't appear to be highly abundant or likely spread through this route. Now, of course, as researchers, we want to address all these questions thoroughly. But I'll spend the majority of the rest of my time, there we go, talking about this idea of using wastewater surveillance or wastewater-based epidemiology to monitor for SARS-CoV-2 within a community. And so the concept behind wastewater surveillance, which is what I'll call it for the rest of this talk, even though I'll use those terms interchangeably, is really very simple, perhaps deceptively simple. And the idea is that because infected individuals excrete the infectious virus or excrete the viral RNA in their stool, at some point down the sewer shed, we can monitor for that virus or the viral RNA, right? And then use that signal, whether it's a quantitative measurement of the level of the virus, or perhaps just a qualitative plus minus detection, to direct some other public health response, whether that be direct clinical testing or other control measures to limit the spread of the virus. And I think that there's really three main potential applications that we could use this for, and it's important to separate these out because I think they sometimes get sort of intermingled. So the first and probably closest to application we have today is using this for surveillance, where we can simply measure, is SARS-CoV-2 present within a sewer shed? Now, there's some important questions that go into this. So for example, people move around between sewer sheds. Not everyone who's infected sheds the virus in their stool, for example, but this is a really potentially powerful application of this approach. The second approach may be trends in infections. Do infections appear to be increasing within a community? And some recent papers that I'll reference seem to show that we can use these concentrations for trend analysis. And the third application that I think we're furthest from actually realizing is using these measurements for quantitative monitoring, or to perhaps oversimplify, how many people within a sewer shed are infected. We really aren't quite there yet, but of course, that is perhaps a goal of some of these research efforts. So like I said, I think that this approach might be deceptively simple, and given some of the other challenges with asymptomatic disease spread and limits in clinical testing availability, I think it's really caught the media's attention in addition to researchers. And you can see just some of the media reports pulled down here. I think the way that this is reported in the media often has something to do with something along the lines that we're monitoring an entire community. And I think most of the people here would recognize that, in fact, is probably not true, right? We're monitoring the sewage for an entire community. And there's lots of research questions and significant technical challenges that remain to realize the full benefit of wastewater surveillance. So I'm going to give just an overview, and this is my opinion, of where are we today in the state of the science. There have been widespread reports of community level detections of SARS-CoV-2 in wastewater. I'm just going to highlight some of the earlier ones here. I think the first one that report that I was aware of came from Gertjan Medema's group in the Netherlands. And this is a table from the preprint and a figure from the actual publication, both citations below, showing successful detections, in fact, prior to some even clinical diagnoses in some locations, and a correlation between observed concentrations of the virus in wastewater and cases within that sewer shed. These work has really continued and expanded worldwide. I think one of the first peer-reviewed papers came from Morish Ahmed's group, and our group collaborated with as well, showing successful detections and sequence-based confirmation of those amplicons in Australia. And another really cool visualization, and hopefully Raul wasn't planning on presenting this later. Sorry if you are, Raul, but I think this is so cool. I wanted to show this. Came from Raul Gonzalez's group at Hampton Roads Sanitation District, and the citation here is below, showing increasing and decreasing concentrations by color here in this animated GIF. Right, and so this has been done really across the world. There's been lots and lots of reports. And I think I could summarize what we've learned so far with a couple of key takeaways. This data is inherently noisy, and I think that that's likely due to variabilities within sewer systems, viral shedding, and perhaps other factors as well. Some studies have suggested correlation with clinical detections. This is typically retrospectively done after smoothing, so it's not done immediately after a detection. And studies appear to vary sewer shed to sewer shed that show that the system parameters are likely important in making some of these interpretations. So far, there's been fairly limited public health actions from these community level monitoring efforts. And I think that there's a clear need for the science to move beyond reported detections and find a way to address some of these challenges with noisy data and how to make this data more actionable. So I'm going to highlight some of the key emerging areas that I see moving beyond these community level detection reports. So I think many groups, including ours, have spent a lot of time on developing the methods behind some of these monitoring approaches. And there's a lot of clear need and uncertainty around some of these methods and approaches. So some of these that are highlighted here, and this list is far from exhaustive, is appropriate sample concentration, processing, and storage methods. There's a lot of uncertainty here, and I'll show this figure again, around shedding of the virus. And that has both to do with the concentration of virus that's shed in stool, and also the length of shedding. There have even been reports of individuals who have shed the virus in stool after being clinically negative by a nasal swab, for example. How do we deal with this perhaps prolonged shedding for some individuals? Where should we sample? This is a figure here of a wastewater treatment plant where some folks are focused on sampling primary solids, for example. Others are focused on sampling primary influent or primary effluent. And many groups are doing this in slightly different ways. Yeah, what's the most appropriate way to sample that's representative of a system, in fact, as well? So there's lots of these important methods questions that are necessary to address some of these uncertainties and advance the science. A really cool idea that I have seen recently is that of alternative detection targets. So this is a report out of Canada showing successful, and this is a preprint, but showing successful detection of the N protein And there's many more copies of these capsid proteins than there is of genome. There are 50 N proteins in a SARS-CoV-2 where there's only one genome. And so this has the potential to increase detection limit or maybe expand detection tools here as well. There's other molecular methods that folks are exploring as well. And finally, I think one of the ideas that's gotten significant attention as of late, but maybe has not quite made it into the literature yet, is this idea of moving up the sewer shed to sample perhaps either smaller subsets of a community or maybe even building level monitoring for the coronavirus. And this is some pictures of our group doing this on the Notre Dame campus and trying to develop some of these approaches. And I know that there are many other groups doing this as well. The basic idea behind the motivation to move up the sewer shed is that sampling at a smaller spatial resolution may improve what you can do with the data. So for example, if we see detections at the level of a community of 50,000 people, it's perhaps not clear today when we know that this virus is fairly widespread, what we can do with that information. But if we have detections in a building with 100 or 200 people where we don't know of any infections, perhaps we can ramp up clinical testing of that facility. And this, of course, could be potentially powerful, not just for college campuses where we're developing it, but also other facilities like long-term care facilities, nursing homes, correctional facilities, or other facilities with vulnerable populations. There's a couple of key points here that I think there's research needed to address. Of course, the data gets noisier, right? So it's more heterogeneous of when people are actually using water, using the sewer system. There's increased sampling challenges. There's a grab sample appropriate. Auto samplers are quite expensive or have their own operational issues. And also there's a decreased need, what I would say, for a quantitative readout. For most of these applications up the sewer shed, I think it's really, the question is whether or not the virus is there, not are levels increasing, most likely. And so for some of these applications, again, I don't think they've made it to the literature yet, but I think that this is where people are realizing we might be able to move public health action a little bit more rapidly. So this is my summary of where I see the state of the field. There have been many reports of SARS-CoV-2 RNA detected in wastewater globally, and a reminder that this is not correlate with infectious virus, and the infectious virus decays quite rapidly in wastewater. The methods surrounding this are rapidly evolving, and there's many groups working on developing these methods. I think interpretation and application of some of this information at the community level to manage public health is more challenging, and that's maybe the next frontier for some of the science. How do we actually use this information? And the most promising approaches are probably these surveillance and leading indicator, perhaps trend analysis applications for individual communities. I apologize, I have to keep moving the screen so I can read. Again, some challenges to reiterate. Wastewater and sewage infrastructure are highly variable, both temporally, day to day, we see relatively high variation during some of our monitoring efforts, and across, of course, location to location. We actually don't know the state of SARS-CoV-2 in wastewater, and like I noted that there have been some reports of viable virus in wastewater. And I think that the most critical outcome of this, as far as advancing the science, is the fact that there's enhanced biosafety restrictions currently in place for handling some of these samples. And if we could address some of this uncertainty, perhaps we could reduce some of these biosafety restrictions. There's some significant trade-offs in not just concentration methods, but a lot of the methods that are applied in cost, complexity, and human time necessary to process them. And so how can we make this data available more rapidly at a lower cost, but also have high quality data as well. And the field has really yet to arrive at a consensus on methods. So for those of you that might be tuning in with an interest in getting into this field, but uncertainty around all of the different reports you're seeing, you're not wrong. There's certainly a lot of variable methods out there that run the gamut from sampling all the way to data analysis. Okay. So I'll begin, or I'll close with just a couple of highlights. I think that this field, at least for those of us that are in water microbiology, has moved faster than anything I've ever seen or maybe even imagined. And I think many of us early on have recognized that collaboration is really key to advancing these tools and making them relevant during the time period of this pandemic. And so there's a couple of efforts to highlight here. So one of these is this WBE collaborative. This was put together by Aaron Bivens, again, who's a postdoc in my group, and the link is here. It's an open door collaborative, Slack protocols, all sorts of things for those who might want to get involved. And I think there's at least 800 individuals who are now currently on this. And so if you're interested, I'd recommend checking this out and seeing if it's useful for you. There's also, and this is available for those that are outside the US as well, a NSF funded what's called Research Coordination Network on this topic. And I'm the PI of that effort. And co-PIs are Ali Baim at Stanford, Heseth Delgado Vela at Howard, and Rolf Halden at Arizona State. And there's lots of activities that are going on through this RCN that I hope that you'll have the potential to find useful. And the nearest one that's coming up is a seminar series that's on November 4th. You can see this recorded here, and the link is here, and this is being recorded, and it's also available at the collaborative website that I just mentioned as well. So with that, I'd like to thank UNC again for hosting this session, and Joe primarily for putting it together. Contact information for me is up here if anybody would like to reach out with any questions or further discussion, and a link to the collaborative that has a lot of available resources. And then of course, thanks to the funders who funded all of this work. So thank you. And then I forgot my next job is to introduce the beginning of the lightning talks, and that's Andre Badia. So take it away, Andre.
Yeah, thank you very much. Hello, everyone. My name is Andre Padilla, and I work for the Costa Rican Water and Sewage Authority. The first COVID-19 case reported in Costa Rica occurred in the beginning of March 2020. Since then, many efforts have been made to halt the progression of the disease into the population. Within this context, SARS-CoV-2 wastewater-based epidemiology approach emerged as a tool to support managed decision by public health authorities. Wastewater surveillance began in April 2020, prior to the community transmission. Next slide, please. However, in Costa Rica, only 23% of the population is covered by sanitary sewer, and only 15% of the wastewater receives at least primary treatment. Thus, creative sampling solutions and strategies were required to monitor SARS-CoV-2. So far, we have sampled a variety of different sites, including large septic field system associated with clinics, hospitals, and airports, surface water receiving wastewater from informal settlements, points in the sewer, as well as all of the government-owned centralized sanitary sewer treatment plants and pumping stations. Next slide, please. Points are chosen in collaboration with public health authorities, and factors like COVID-19 infection rates, syndromic surveillance, economic indexes, and suitability for sampling are considered. Composite samples, as well as small volume grab samples, are concentrated and analyzed by an adsorption extraction techniques, and using RT-qPCR with CDC assays for target N1 and N2. The reports are immediately shared with the Ministry of Health and to the Social Security Fund, which is our socialized medical care system. For us, the sampling process has been the most challenging aspect in executing wastewater analysis. We have access to 2 auto-samplers and have limited personnel and funds. Additionally, the challenge related to heavy rains in the tropics are affecting all our sampling locations. So how are we overcoming these obstacles? Next slide, please. More swabs reemerge as an affordable and effective sampling approach to overcome our sampling challenges. Our laboratory has extensive experience using these affordable devices from the Latin American outbreak of cholera in the 90s into the regular surveillance of Salmonella typhi in surface water. So we quickly adapted this technique to concentrate and subsequently detect SARS-CoV-2 in wastewater. Basically, we make the swabs by putting together 3 pieces of cotton gas, folded in thin several times until form a roll, and then a strong wire is tied to the center. And that wire is used to attach a nylon fishing line, which is used to suspend the swab in the water or the stream. Next slide, please. In our experiments, swabs were immersed for 24 hours, and elution was done by adding PBS and hand massaging the swabs. Eluates were then centrifuged to remove debris, and both supernatant and pellet were analyzed for SARS-CoV-2 and PMMOV. And in comparison with composite and grab samples, Moore swab was more likely to detect SARS-CoV-2 in higher concentrations, and PMMOV was also found in higher concentrations. Therefore, Moore swabs have been a practical tool in when relative quantification can be used in decision making. Next slide, please. Despite many challenges, wastewater surveillance has served as an early alert of increasing infections and guided community level testing efforts in Costa Rica. The implementation of a national surveillance system require an interdisciplinary group of institutions, and improving communications and establishing clear roles and responsibilities are fundamental to interpret and make decisions based on all data, both traditional and those derived from wastewater analysis. Thank you very much.
Thanks, Andre. Questions, please put them in the chat. We have a couple of minutes for questions. I actually wanted to ask Andre, a couple of people have asked about, non-wastewater matrices like sludges or, other something that waste flows that are different, maybe in concentration than you would find for wastewaters. Is that something that your group is doing? And do you have any thoughts on that?
Yeah, we haven't been able to test many things that we would like to. At the beginning, we tested some primary sludges, and it gave. I mean, when we knew that we're positive result there, it showed good results. But based on the fact that we have personal limitations, so we are just focusing on wastewater.
Got it. Okay, let's move on, please, to the next talk. Next is Raul Gonzalez of the Hampton Roads Sanitation District. So if you wouldn't mind, Mary Margaret, please. And Raul, take it away.
Oh, thanks. Thanks, Joe.
Yeah.
Is my presentation up?
Doesn't appear to be. There we go.
All right. Hello, everyone. My name is Raul Gonzalez. I'm an environmental scientist at Hampton Roads Sanitation District. And I'll be quickly sharing some of the data, some of the work we've done to compare grab and composite samplings at a wastewater treatment facility. Next slide, please. So we know that raw influent flows fluctuate continually at the headworks of a treatment facility. What we don't know is if these fluctuations correspond to significant SARS-CoV-2 concentration fluctuations. And so early on, we had the specific question of do grab samples sufficiently characterize wastewater SARS-CoV-2 concentrations, or are flow-weighted composites necessary? Just as a little background, for permit applications, I think we at a treatment facility we normally do grab samples, but for a project like this, for a new project like this that involves molecular samples, I would normally just immediately go to a 24-hour flow-weighted composite for a representative sample. But with the amount of sampling that we're doing, we don't have enough composite samplers to do this. So that's where this question kind of came from. Next slide, please. So as we were thinking about that question of grabs versus composites, as we were just talking that out, we thought very simply, if the variation of grab samples over time doesn't vary very much and it's not that different from a composite sample, then we would say, okay, we could use grab samples in place of composites, and we'd feel good about it. So to address this question, we did a really small study at one of our treatment facilities, our Army-based facility, where we took grab samples every 2 hours for 3 days straight, and we also set out concurrent 24-hour flow-weighted composites. So on the bottom of this figure, you'll see that during the study time, the flow at the treatment facility varied from 7 to 17 mgd. And on the top of the figure, what you're seeing is, so in the 3 colored lines, you're seeing the results of the 3 SARS-CoV-2 assays, N1, N2, and N3 over time. At the bottom of the figure, and the units of that are log copies per 100 mil. And at the bottom of the figure, you're seeing the concentrations for the 24-hour composites of the 3 assays. So without looking at any numbers, just qualitatively, the grab sample concentrations if you look at it per day, don't vary that much compared to and don't really change that much from, or don't deviate far from like the composite concentrations. You are seeing these like dipping concentrations where that correspond to low flow times. But overall, I would say just by looking at this data, if you can take grab samples, you should probably take them around peak flow or right around there, and avoid times that are low flow conditions. So I have some stats here on the right, but really, I really like to look at this coefficient of variation, that's 69% of our grab samples. That allows me to compare it to different data sets and really put that variation in perspective. I was trying to figure out how to interpret this 69%, and just this morning I thought, oh, I should compare that to the coefficient of variation of all of the Army-based data so far, and that's 180%. So that's coefficient of variation is 180% for all of our weekly sampling at that facility to date. So kind of in my mind, I'm thinking, okay, then that's relative to that weekly sampling, this is actually a lot smaller. Next slide, please. I'm actually going to slip. This is an old slide set, so next slide. So what about the rest of? So what about other other viruses and bacteria besides SARS-CoV-2? So in these exact same samples, we also enumerated a bunch of other pathogens and indicators. So on the top left, those are the exact same samples. So across the board, I couldn't show all the data, but across the board, seeing even better agreement in the grab samples. So concurrently with this study, we actually also monitored a pump station, so farther up in the sewer shed, and that's what you're seeing in the bottom right table. And if you look at that coefficient of variation for those assays and compare them to the grabs in the upper left table, you'll see that there's more variation in the upstream samples than at the treatment plant samples, which was mentioned, I think by Kyle Bibby. Makes sense. And I have a feeling that if you were actually do this farther upstream, you probably see a little more variation there. Next slide, please. Oh, you know what? That actually might be it on this slide deck. So with that, I'll take any questions that anybody might have. Don't know if I went fast there.
That was perfect. Anybody who wants to ask a question, please put it in the chat.
Jill, I think the chat's disabled.
I think people are, the Zoom chat is disabled, but the Pathable chat is active. Yeah.
It's under poll for those that might be a little bit unclear.
I'm not sure if it is under poll. It's under, there's like a chat tab on the main Pathable meeting website that people have been submitting comments to. So I'm not sure if everybody's able to access it, but it's on the main Pathable conference site for this session rather than on the Zoom. I hope that's clear for folks. Okay, well, as you, okay, so Aaron Bivens asks, what was the COVID-19 prevalence at the time of sampling? And someone else asks, did you communicate your findings with the Army personnel? I guess those are both for Raul.
I think in that service year, we had about 200 cases, maybe a little bit less than that actually, because that's for the city, but that city actually has 3 different facilities on it. And did we, so the treatment plant, so what's interesting is that those SARS-CoV-2 results were for the treatment facility, but the pump station that we monitored farther upstream was actually specific to that base. And the reason I didn't show any SARS-CoV-2 data for that was because we saw non-detects at that facility. So it actually wasn't coming from that Army base, it was from another part of the service area. But all of our data does go to the health department, and there's various other stakeholders that want to be kept in the loop on treatment plant data. So we do give that to them as well. You can also see updated data on HRSD's website. We post our updated figures.
Okay, let's move on to the next presentation. Thanks a lot, Raul. And I'm noting questions down, by the way, for folks. If we don't have it for time for individual speakers, we'll collate these and put them to the panel in the panel discussion. Next speaker is Hannah Greenwald from UC Berkeley. So Hannah, please, the floor is yours.
Great, thank you. Hi, everyone. I'm Hannah Greenwald. I'm a PhD student at UC Berkeley in Cara Nelson's lab group. And I'm here to talk to you guys today about our work on wastewater-based epidemiology in the San Francisco Bay Area. So next slide, please. So when we first started this project at the beginning of the pandemic, we were pretty new to it, as are a lot of us. And we started off by working to develop methods to detect the viral RNA in our sewage. So we developed this method, which we call the sewage salt, silica, and SARS-CoV-2 method, or the 4S method for short. And it's a direct extraction method in which we're inputting 24-hour composite samples of raw wastewater. So you can see here in our first panel, we take the wastewater and add a high concentration of salt to it to lyse the virus. We then heat and activate it, and then filter out the solids. We then ethanol precipitate the RNA, pass it through a silica column, which you can see in the middle panel here, and then wash the column to clean the RNA and release it from the column. We can then input that into molecular methods. In our lab, we're using RT-qPCR, but you can also do RT-ddPCR. And so far, we've really liked this method. We've tested it against commonly used ultrafiltration methods and seen higher recovery. And it's kit-free, which has helped with some supply chain issues that I know a lot of other groups have been having. And And yeah, so a few other groups have also adapted this protocol, and you can find it on protocols.io. Next slide, please. So using this, we've started monitoring our wastewater regionally. We started up a regional wastewater monitoring program called COVID-WEB, or Wastewater Epidemiology for the Bay Area. And this work involves many stakeholders. So we've involved public health officers from across 6 counties, wastewater agencies and universities, being UC Berkeley and Stanford, all to help contribute to this work and making what we're doing actionable. We've also started a regional monitoring lab out of our UC Berkeley campus to process all of these samples, and we try to get them back within a 72-hour turnaround time. So you can see here maps of the regions where we're currently collecting samples. So in Marin or North Bay, we have 7 regions, 2 targeted subregions, and then we're also sampling at facility scale at San Quentin Prison. In the middle panel, you can see San Francisco. We're targeting 6 regions, and we're also targeting facility scale skilled nursing facilities. And then in Oakland or Berkeley or East Bay, we have 3 sub-sewer sheds, which I'll highlight further in a minute, and also campus sampling at our UC Berkeley campus. Next slide, please. So here you can see the 3 sub-sewer sheds in the East Bay. And I wanted to highlight just a few of the results we've been getting and how we've been trying to analyze and interpret the wastewater results that we're processing. So here are highlighted our results from the S68 sewer shed, which represents Oakland. On the top, we've plotted case data from diagnostic testing, and on the bottom is our normalized SARS-CoV-2 data from our wastewater samples. And we're looking at the N1 gene normalized to PMMOV, or pepper mild model virus. And we can look at these results over time and see that they're tracking with the case data with a slight lead time. Next slide, please. And we can also compare regions. So you can see for our 3 sub-sewer sheds here, plotted on the left is our new COVID-19 cases per person per day. And then on the right are our wastewater results. And you can see that the A sub-sewer shed has the lowest prevalence in both cases. The S sub-sewer shed has the highest. So wastewater data could also be used to kind of assess burden of infection in communities. Next slide, please. And then lastly, I wanted to highlight our one of our sampling from our facility scale. So here, University Village is a housing community on the UC Berkeley campus with about 2,700 residents. And we were able to detect a spike where we detected presence of SARS-CoV-2 in the wastewater coming from this housing community, and they were able to bring in more diagnostic testing. We were able to know that there were 2 diagnostic cases reported for the associated spike. Next slide, please. And with that, thank you.
Thanks, Hannah. Question, what's the amount of lead time of cases from the Bay Area data?
So we're still going back through a lot of the results I presented are archive data from the archive samples that we were freezing this whole time. So we haven't processed it for all of our sub-sewer sheds yet. Just for this one, it's looking like a few days, but we still have a lot more analysis to do there to better compare it to what other groups are coming up with.
And let's see, will the COVID-WEB data be made available to the general public? Is it on a public-facing dashboard of some kind?
It is not currently on a public-facing dashboard. Right now, we're just sending it out to public health officials, and we're still in the works of deciding like what the dashboard would look like afterwards to see if it might become public-facing, but right now, no.
Got it. Okay, thanks, Hannah. Appreciate it. We're going to move to the next speaker. So Catherine, the floor is yours.
Great, thank you. My name is Catherine Hoare. I'm a postdoc in Andrea Silverman's lab at NYU. And in partnership with the New York City Department of Environmental Protection and some other collaborators, we've been working on the development of a monitoring program for SARS-CoV-2 in the city's wastewater. Next slide. So routine sampling for this program is completed twice a week through the collection of composite samples from the city's 14 wastewater treatment plants that are serving the 14 sewer sheds that are shown here. I've pulled out on the right two different sewer sheds results from the end of August to about mid-October. On the left axis, the black squares, we're looking at a normalized amount of SARS-CoV-2. So this is concentration of N1 gene copies in a sample normalized by the flow rate and the population within that sewer shed. And then on the right, the red line is an estimate of the number of cases per 100,000 people in that same sewer shed based on publicly available data. So what we see for both of these sewer sheds is this sort of low level amount of SARS-CoV-2 in the wastewater during the late summer and early fall when cases were also low. And then for some areas, like the sewer shed in the top graph, we're seeing a bit of an uptick in the wastewater data. And these are in places where we're also seeing increases in cases in some areas. Okay, next slide. So one of the major goals for this program from the beginning was to set up an entirely in-house system for the DEP. So that means that every step of this process is happening in a DEP lab. So this is the workflow the DEP is selected, developed in cooperation with a team over at CUNY. A quick overview of the process. Influent samples are pasteurized, solids are removed, and the supernatant is filtered prior to PEG precipitation and extraction of RNA with a QIAGEN kit. And then we're targeting the N1 gene with a one-step RT-qPCR assay. So as Kyle mentioned, there's really no solid consensus about methodology. There are a lot of possible combinations of different approaches that can be used. And in starting out with this, when we were looking at and assessing different options, what we were finding is that, you know, what was working really well for one group wasn't necessarily working well for us. So this made us really curious, obviously for finding an optimal workflow for the city, but also in thinking more broadly about why some of these steps helped or didn't help in optimizing the signal we were getting for SARS-CoV-2. Next slide. So we've been looking at a few of these steps, a big one being pasteurization. So initially, like a lot of others, we actually eliminated pasteurization based on the large decrease in signal we saw for bovine coronavirus after pasteurization. And so bovine coronavirus was the process control that we're spiking in. But what we've seen for SARS-CoV-2 signal is that it can actually be increased with pasteurization. And this is consistent with what others are also reporting. So I'm just showing here some preliminary data of a comparison of the N1 gene concentration for 4 different plants. Here it's on a log scale. So these are 4 plants in New York City, and the samples have either been pasteurized in the black circles or not pasteurized in the gray squares. Otherwise, these samples were treated exactly the same based on the workflow I showed in the previous slide. So across the plants and the different concentrations that we've looked at so far, for the selected PEG method that the DEP is using at least, we're seeing this increase in measurement of SARS-CoV-2 RNA after pasteurization of the influent. Next slide. So this is something we've started to look at a bit more deeply, along with some other steps, to try and parse out the mechanisms behind these differences and understand why this is happening. So for pasteurization, for example, some of the hypotheses we're looking into is that heating might help disaggregate viruses from the wastewater solids, or perhaps heating helps degrade inhibitors to RT-qPCR. And then finally, another component of our ongoing work that Kyle also alluded to is characterizing this variability in the data. So the data is pretty noisy, and there's a variety of reasons for that. One of them, particularly for our program, to think about is the fact that there are different technicians doing this analysis every day. So part of this work is looking at the data from the summer, which we can kind of think of as a baseline when cases were consistently low, and use that to help understand the variability and get a better sense of how to set thresholds moving forward. Thanks.
Thanks, Catherine. I'm glad you brought up data smoothing. That's and Kyle did as well. I mean, I think that's a huge that's a huge issue, and we need to figure out what we can do with noisy data. A question from the chat: what's the starting volume of wastewater prior to starting the pasteurization in advance of extraction that you used?
Yeah, so we're starting with 40 mil of wastewater.
Great. Okay, all right, thanks. Thanks, Catherine. Our next speaker is Amy Kirby of CDC. Amy, the floor is yours.
Thank you, Joe. Let's see. We have slides. Excellent. All right, so what I want to share with you today is a brief overview of the National Wastewater Surveillance System, or NWSS, or NEWS. And this talk will really focus on a programmatic overview of that system. But the technical underpinnings of the system were posted on our website on Friday. So if you're interested in those, I encourage you to go look them up. You can do a quick search for CDC and NEWS, and it will take you to our landing page where you can get to all of those other pages. Next slide. So I think all of us are familiar with this, but I just wanted to give a quick overview of the four main reasons that we're really focused on wastewater surveillance as a useful tool for disease surveillance, even in the setting of intensive clinical surveillance as we have for COVID. So the first is that it's a very direct measure of community infection levels, which we don't have coming in from our clinical testing. Second, it captures asymptomatic and pre-symptomatic cases, which none of our clinical surveillance can do in a systematic way. And third, and what I think is really the most important advantage here, is that it's not influenced by healthcare seeking behavior, healthcare access, or healthcare testing capacity. And that's particularly important in a setting of these explosive, fast-moving pandemic where we see healthcare capacity get overwhelmed. And finally, as many others have said, the time from changes in infection to available data can be quite short, measured in days rather than weeks, as we see for some of our clinical surveillance. Next slide. So this is a schematic of the NEWS system. There are three primary stakeholders in this system: wastewater utilities, laboratories, and public health departments. CDC's primary focus is on those public health departments because they are the end user of this data. If health departments aren't fully engaged in this process, none of this data is going to be used for a response. So our team is working to build capacity within those health departments for coordinating wastewater surveillance and integrating the results of that surveillance into their COVID response. We've also partnered with the Water Environment Federation and the Association of Public Health Laboratories to provide similar dedicated support to the utility and lab communities, respectively. Of course, in any surveillance system, CDC's role is really to ensure that the data is comparable regardless of where it's collected. And that's really important for wastewater surveillance. So the NEWS data portal can accept data generated with any of the common methods that are being used. And the analytic engine can then provide standard data analysis that can adjust for assay performance and normalize for fecal input. And those results are then provided back to the health department in a standard reporting format. Next slide. There are 15 jurisdictions that are using CDC funding to support wastewater surveillance activities. 8 of these early implementer jurisdictions, identified in blue on this map, were identified based on their capacity across all 3 stakeholder groups for wastewater surveillance, so utilities, labs, and public health departments, as well as their willingness to engage in the development of a new surveillance system at this time when they're juggling so many other responsibilities for the response. An additional 7 jurisdictions, noted here as red asterisk, chose to redirect some of their earlier COVID funding from CDC to support wastewater surveillance. And among these 15 different jurisdictions, we see quite a variety of approaches in these jurisdictions. Some of them are aiming for % population coverage, some are targeting high-risk areas, some of them are focusing on a few pilot sewer sheds, and many of them are including this targeted use surveillance or upstream testing. So looking at either subsections of a sewer shed or testing individual facilities like prisons or universities. Next slide. The primary analytic result for NEWS is a trend assignment for SARS-CoV-2 RNA concentrations for each sewer shed. So on this time series graph, the SARS-CoV-2 RNA concentrations are plotted as black dots. The gray bars are case data that's restricted to the sewer shed population and plotted by date of symptom onset. And both of those data points are presented as rolling averages in the black and gray lines. But I want to draw your attention to the horizontal bar graph at the bottom. Our system uses linear regression to assign a trend, either growth, decline, or plateau, based on the 3 most recent measurements. And if a trend continues for 5 or more measurements, it's considered sustained. So you can quickly see by looking at this, that this sewer shed was doing well in late April and early May, shown in green and gray, but concentrations started rising before Memorial Day and have stayed high through the summer. Next slide. So as others have said, although wastewater surveillance has great value, it also has limitations. And I want to point out 3 that are particularly pressing for us at CDC. First, we don't understand the overall surveillance limit of detection. So how many people need to be infected in a community before we can reliably detect viral RNA in the wastewater from that community? Second, like all environmental pathogen tests, negative samples cannot be interpreted as the absence of the pathogen. And so we can't use wastewater testing to clear a community. And communicating this to end users is really a big challenge if they're used to clinical testing where a negative means you are not infected. And third, the resources for this testing are limited. The test is inherently not throughput. There's a limited number of labs that can handle this testing, and frankly, almost all of them are on this call. And we're seeing shortages in the required sampling and testing supplies. And so with all of those limitations, we're really not able to implement wastewater surveillance everywhere right now. And so we need to focus our implementation on places where we know it will provide the best information right now and scale as resources allow. Next slide. I cannot end without acknowledging the incredible team that stood up an entirely new surveillance system in just a few short months. And if you'd like to reach the news team, you can reach us at that email address, eocevent456@cdc.gov. Thanks.
Excellent, Amy. So important. You know, we need to be, I think, as a community, really trying to figure out how we engage with the public health community. And so I hope we can discuss this more in the panel discussion. Couple of questions. One person asked for the link that I think you mentioned near the beginning of your talk. There might be some utility to putting that in the chat on Pathable.
Yeah, I think you shared it.
Oh, I tried to find it, but I don't know if that's the right one. I think I did.
It is. That's going to be our landing page, and then all of those technical pages are linked from the bottom of that page.
Okay. And Colleen asks whether there'll be a public-facing dashboard for data for wastewater surveillance.
Yeah, that is the goal. So our initial goal is to get this data back to the public health departments that can use it. But we do have a goal of getting a public-facing dashboard. It will be part of the COVID data tracker that's on the CDC website. And we're hoping to get that live in the next few months.
Excellent. Thank you. Thank you, Amy. We'll move on to our next speaker now. So Manish Kumar of IIT Gandhinagar, the floor is yours. Thank you for joining, and doubly thanks because I know this is not a great time for you to join us from India. So thank you so much.
Thank you, Joey, and thank you, Kyle, for inviting me. Yeah, I would go ahead. Can you go to the next slide? Initially, there will be a little bit problem in the animation because I improved later, but now it is. So please, next slide. What I am going to say about is that the whole entire experience in last 7 months or so. And I would congratulate Arun and Kyle and also the many who has really initiated this this global collaboration. Of course, this paper took a long time, but still it was the first initiation. Next. Next. So and then gradually the things were getting recognized and several other paper came. Next. And then, of course, we could manage to announce our success by May, by June first week or something. And next. And then, of course, but what happened is that, and then this surveillance system has given a lot of new information. Next. Like it has come to the March 2019 rather. So these were some of the strongest support that has got wastewater surveillance. Next slide. Next. And then, of course, we have, because I feel that water connects life and environment. And that's why this all has to come into the water. And what we are talking about is wastewater. Kyle is also a co-author here. And we have given a holistic pathway of these viruses. And of course, that strengthen the wastewater surveillance. Next. Of course, it is very strong support that testing a country like India or South Asia, where there is a very a thick population, individual testing may not be enough. And that's where it was very, very important. But I tell you that there were a national wastewater surveillance system that Amy has proposed, but in India, we could not do that. And for next, but we had some little bit things like in Hyderabad, they come up like, okay, there were many people done. And then recently in Bangladesh also, we could detect the COVID-19 in the wastewater. However, we don't have still a national or even in a province scale surveillance system. Next. Next, go ahead. So what I would do is that I will just talk about 5 issues that Kyle, many of my next slide, please. Many of them have touched upon. Here I just want to, yes, just hold on. So here I wrote ENC1. It means that experience and challenges, number 1, discrepancies among the monitored gene. If CDC system and TechPath, we monitor more than 3 genes, N gene, S gene, and ORF gene, but we don't know that which gene has to be taken into account for the final estimation of total RNA. Because the point is that whether one gene detection is enough, or of course, my friends have shown that N gene normalized by PMMOV and something like that. But here the point is that whether we should go with the average or the maximum. But here, this 2 months, 3 months analysis of our data shows that it doesn't follow N gene, S gene, and ORF gene if talking about intact RNA. So that is the one question. Next. So question number 2 is that what about the treatment plant? What is the role? Does treatment removes it all? Of course, I have seen in plenary section that, okay, treatment does it. But initially, we also reported that there is a total removal. But recently, we are not finding a significant removal. This is a case where it is 10 MLD plant and non-significant removal we have noted in N genes. Next slide. But when the plant becomes shorter, this is a campus 2.3 MLD plant, and which is equipped with ultraviolet system or micro filtrate system. So here also we have found that gene was detected in the treated water sample also. Next. So the challenge number 3rd, that is next slide. So challenge number 3rd is that whether the genes accumulate or dilute in the sewer network system. I support or don't support, I don't know, but the Kyle is saying that we should go upstream to the sewer system. But here what we have found is that in Bangladesh, we took the sample from a detention site, and we saw that secondary and tertiary, the tertiary drains has shown the accumulation of the genes. Go next slide. Next. Yeah, so this is a challenge number 4, and then I have only 1 slide, so I will end up. This is about that, do the count of genes in the influent, how much genes are present in the influent is going to determine the effluent or not. Next slide. So here, if you can see that we had reported that there is a total those things, but several, even in China and Spain, it was reported that after treatment, there were the genes detected. Next slide. Okay, now this is very critical. Kyle has highlighted, Hannah has highlighted, Amy has highlighted that what to compare with, whether we have the data of confirmed people, active people, recovered people, but we know that even recovered people will shed. A person which is not confirmed, still if they will shed the genes. So when they are going to, when we can see that, okay, this is what they are doing it. So what to compare with the actual number, because Kyle has pointed out that number of exact patient in the hospital. But here, if we take the data from the public offices, or to what data we should report. So here again, I would tend to agree with Kyle that we have to have a smaller scale, it can be correlated. Here we have found this 2 weeks lag more between the gene and then the given population change. But sometimes we also find 4 weeks. So 2 to 4 weeks early warning is possible. And with this, this is the 5 challenges. I just brought it to share with all. And so thank you very much. Next slide. So this is just a summary, is that for India, when we talk to the government that why we cannot start this surveillance, they say if COVID will go, what genes we will keep on this one. So that is also one of the points. Of course, frequency of sampling is there, what to sample, where to sample, only STP or open range, and so on and so forth. Of course, I don't want Joey to say stop talking, and that's why I want to end it. Next slide. Just I need to say thank you to my funders. So next slide. Yeah. So this is a project supported by UNICEF and the Kiran C. Patel, and of course, thank you very much.
Thank you so much. Really interesting. Let's, in the interest of time, let's move on to our next speaker. We'll come back to some of these themes in the discussion, I hope. Pengbo Liu and Christine Mo, I think you were next. The floor is yours.
Pull up our slides. All right, thank you. So my name is Christine Mo, and my colleague Pengbo Liu and I have been working on COVID wastewater surveillance at an institutional level on the Emory University campuses. So today we'd like to tell you how we have been using more swabs as a sensitive and low-cost approach for collecting composite samples to test for SARS-CoV-2 RNA. Next slide, please. The goal of this surveillance is to provide early warning of COVID cases in residence halls on Emory campus. So for this situation, we are interested in a presence-absence result, as Kyle alluded to in his opening remarks. A positive signal in the wastewater is followed up by a public health response, so repeat sample collection and analyses, symptom surveillance of the building residents, and diagnostic testing. Collecting and testing the samples from the residence halls is different than samples from influent at a municipal wastewater treatment plant. The catchment population for these buildings is small, about 37 to 185 residents per building, and the wastewater flow is variable. We first started collecting grab samples from manholes directly downstream of the residence halls, but we soon found that many manholes were not well suited for grab sample collection due to low flow, and some of them are very deep. Also, we wanted a method to collect a composite sample rather than a brief sewage snapshot. So Andre has described the more swabs already and their use for cholera. We have been using them in Kolkata, India for the past 2 years for typhoid environmental surveillance. So we decided to try them for our campus surveillance strategy by placing them in the manholes next to the residence halls. So now I'm going to turn the presentation over to Dr. Liu, who will explain our methods and results. Next slide, please.
So the Mo swab method include 5 steps. First, the Mo swab sample collection from manhole, then Mo swab like processing in the lab, then we did like PEG precipitation, and then RNA extraction, and we run real-time PCR to detect SARS-CoV-2. Next, please. So Table 1 shows SARS-CoV-2 detection on Emory campus. So we collect the Mo swab samples from 3 Emory campus, so main campus, Claremont campus, and Oxford campus, and 2 buildings. So quarantine building is a building where the confirmed students' cases are in quarantine. Emory Hospital is a building where the COVID inpatient stays. So we have numbers of the manholes we collect a swab from and how many swab we tested so far. We can see that the positive rates are very low on Emory campus. So the swab positive rates in the quarantine building was 40%, and in the Emory Hospital building was 87.5%. The CT value for the positive sample are between 24 and 38. Table 2 shows that COVID detection in sewage water using more swab in the quarantine building. So we have the total number of cases staying in the building. So we collect swabs from 3 sets, and the SARS-CoV-2 was only detected from the set 3. So that is where the confirmed cases were staying. And the other 2 sets without the cases were all negative. So we got a positive result when there was only 1 case in the building on September 29th. But this was not true on September 22nd. Next slide, please. Table 3 shows the SARS-CoV-2 detection from Emory Hospital sewage. So we only collect grab sample from Emory Hospital sewage line between week 1 and 5. Starting from week 6, so we collect both grab sample and swab sample. So we also have confirmed COVID inpatient numbers from the dates we collect the sewage samples. We can see that most of the samples were positive. So we have a few negative samples, and there were some reasons for that. So I also want to mention that this sample will always collect around the same time on Monday mornings. Next slide, please. Summary. So most swab are a sensitive composite sampling method. It may be sensitive enough to detect 1 to 2 infected people in a building. It is a convenient and low-cost COVID sewage surveillance method. We can use this method for early COVID-19 warning. This method can be used for institution-level surveillance. Next slide, please. So we would like to give thanks to all the people working on this project. Also, thanks for your attention. Welcome any question and comments.
Thanks, Peng Bo. A few questions on this, some of which we may have to move to the discussion in plenary. But a couple of questions, how you process the more swab samples prior to PEG precipitation. And so if you could describe that, and also your thoughts about sensitivity. I mean, it seems like what you've got is we're sort of trying to empirically say, look at whether a sample is positive based on how many people are in that building. Do you have any sense about sensitivity overall, or do you have plans to look for that?
Yes, great question. You first, when we process the more swab sample, I think it's very tricky. So we first collect sample, then squeeze some liquid out of the swab. It's only like 5 to 10 mils. Then next step, we will use like 100 mil of the elution buffer, which has some component to help virus elution. So we just squeeze the water again and also pull this 100 mil with the previous liquid. So we did it again. So we combine those all the squeeze the liquid. So totally we'll have problem 250 mil of the elution buffer with virus. Then we proceed with the PG precipitation. So in terms of the sensitivity of the method, actually before the assay development, so we spec the called BRSV in the water, like tap water. So we did the series dilution. We also spec different amount of BRSV. We test it. We make sure that I think sensitivity we can detect like right now 4 logs of BRSV in 2 liters of water. So we'll continue to do this experiment. Thank you.
Okay, thanks. We'll have to leave the other questions for later, hopefully. I want to ask Jill Stewart of UNC if you wouldn't mind taking the floor and presenting. Thanks very much.
Hi, everybody. Let me share my screen. So I am pleased to be here today. I'm Jill Stewart. I'm a professor at University of North Carolina, and I'll be presenting today about COVID-19 wastewater surveillance in North Carolina. And I'm also representing the North Carolina Wastewater Pathogen Network, or NCWW PATH, which is a coordinated network in the state. And also need to give a shout out to my students who've been working like crazy on this project and who produced a lot of the data and slides you'll see today. So North Carolina PATH is a coordinated sampling of wastewater treatment plants across North Carolina. North Carolina has a long and strong history of water, wastewater, microbiology, engineering, and so early in the spring, there were a number of different groups who were very interested in wastewater surveillance for SARS-CoV-2. And so with thanks to the North Carolina Policy Collaboratory with some funding from the North Carolina State Legislature, we were able to at least to coordinate. So even from the beginning, the different groups who were working on this were coordinating, communicating, having weekly meetings, standardizing methods, and that includes groups at UNC. The coordination is being led by Rachel Noble at Institute of Marine Sciences, but it also includes groups at NC State University, at UNC Charlotte, at UNC Wilmington. This is an old map. It's also Eastern Carolina University. The full group is working, to Dr. Kirby's point earlier, the full group is working with the North Carolina Department of Health and Human Services, as well as North Carolina Department of Environmental Quality. I'm going to show you just a little bit of very preliminary data. First, the workflow. I don't expect you to be able to see all this, but to other speakers' points, the methods are not quite standardized yet. And so early on, we were trying a number of different things that included both wastewaters and solids, direct extractions and filtrations. And long story short, preliminary data shows that the filtrations from what we tried, using an HA filter worked the best. I'll also note that we are not able to filter. I know some other groups within our network and elsewhere are filtering larger volumes. We're not able to do it. I think that's very dependent on the type of wastewater that you are using, as well as the patience of the people who are filtering, but we're filtering about 20 mils currently. And even with those volumes, we're seeing pretty consistent positives in our data. So this is just very preliminary data. It has not been smooth, it has not been normalized. We do have plans for doing all of that. We also have plans for getting better health data. But even with this very raw data, you can see that we have detections. This is the N1 gene in wastewaters. You have variable detection over time. And this one is an example where you can see this going up and down and an increase right before you see an increase in the number of cases. In this case, this was a treatment plant in a college town right before a college opened. You see this increase, you see the amount of viral RNA that goes down just before the number of cases go down, and then an increase when the state started easing their restrictions. So we are seeing some nice trends that align with the clinical testing data. And this is true across much of the network. And so we have data across the NC PATH network. Here's a comparison of, and one of the great things about the network is that we can compare wastewater treatment plants of different sizes and service areas. So you can compare here a plant that's more of a metropolitan suburban North Carolina plant versus an eastern rural North Carolina plant. And you can see for this larger plant, there's consistent detections, a very high detection right around 4th of July, well before the case numbers increased. There was a lag there, and you can see increases up and down. And even in the rural areas, we do sometimes get detections there. And when we do, it does tend to correlate with this increase in cases, which has been interesting to see. I think, all this is pretty raw and we're still working through data. But points that I think that we'll be working on in the immediate future. Level of resolution for the case data, I think, is important. There's a lot of discussion around what case data is appropriate. We're trying to get better case data. In the meantime, we have like the publicly reported data by zip code, which is not ideal and can result sometimes in a little bit more messy graphs. But then this, for example, is a rural treatment plant that's also within the network. The service area is below 5,000 individuals. And the zip code that contributes to that, this is the main zip code that contributes to that, also has kind of lower number of cases. But if you expand that out to the county level, you can see trends a little bit better with increases preceding case rates. However, a lot of this county is not necessarily on septic. It's not necessarily contributing to this treatment plant. So I think there's a lot of questions about what sort of data we want to use and what is the spatial relation of the spatial area where we want to get that data. And so we're putting a lot of resources right now into improving those so that we can build better models. Part of what we're doing is working with space-time modelers at UNC and at NC State. And they're already kind of thinking through ways to interpolate data and make better data. This is an example of a Bayesian maximum entropy method by Professor Mark Sayre, where you can take the data that you have and then build maps over space and time. And then when we can map out the sewer sheds, we can then cut out the data that we need in order to look at associations. And I'm also very excited that in North Carolina, we're linking the sewage data to population cohort studies. So I think trying to think of the spatial and temporal extent of the case counts and where the case counts are coming from, and whether it's 1 day or 3 day or 7 day or how we want to do it. There's lots of those questions, but there's a lot of data quality issues with all of this. And so I'm really excited to be linking to some of these population studies where researchers are going and measuring the prevalence or seroprevalence within a cohort that has been selected and followed and is representative of either their county or the population that is being measured. And so we will have data from those more rigorous cohort studies along with the sewer data in order to build better models and hope to get to that predictive point that Kyle mentioned at the beginning of can we ever collect a sample and start extrapolating out how many people we think might be sick. And that's going to be, it won't just be based on the modeling, it'll also be based on environmental factors and flow and other things, of course. So we're doing a lot of modeling efforts on that. So I will end my talk in this whole session on some of the advantages of COVID wastewater surveillance. They are varied and some of them have been already covered here, but for example, that it's opportunity for lower cost non-invasive disease surveillance instead of having to sample entire populations or even large parts of populations. Potential for early identification of population infection trends. I know this is a little bit held back by how quick we can turn around samples. And I think there's a number of labs who are working on it and increasing that. But the data and the model so far do suggest that you see these spikes before you see the spikes in the population. So I think this can be really useful and has the potential to overcome representative and stigma issues and access issues for people who may not have easy access to sampling or to clinical sampling. And that is it.
Thanks, Jill, and thanks to all the speakers for staying super on time. I can't believe it. So we should all be giving ourselves a round of applause. Before we move on from you, Jill, a couple of questions. Is the North Carolina data being made public?
Good question. So we are working on that. There's a couple of dashboards that we will likely link to. There's one in the UNC School of Public Health that wants to link to the data, as well as NCDHHS. And so we're working through that. And interesting, I think a really important consideration for that is not just we have the data, we want to make it public, but really also working with each of the treatment plants. And so that, you know, with 20 treatment plants within the network, we're going through the stages right now of working with them and wanting to communicate with them as well. But yes, there are plans for that, and there's a lot of discussion around that currently.
Also, are you sampling in septic tanks?
We are not currently in that. So that is something we also talk about a lot in North Carolina. So I think there's a statistic earlier that about 25% of people in the country are on septic. In North Carolina, it's way higher. And we are missing some of these underrepresented groups. I think it could be interesting, but oftentimes, particularly in households where you have 1 or 2 people, it might just be the same to go and sample the people than the septic tanks. So I think it's something to think about, but I don't know if this approach will be the most appropriate for reaching some of those underrepresented populations, unfortunately.
Got it. Okay, thanks, Jill. We're gonna, I think, move, I guess, to a panel discussion here. We've got about 20 minutes. And if all the speakers could just unmute yourselves. I'm going to try to synthesize a few of the questions that we've gotten on chat so far and put them to some specific people. We've had a lot of questions on methods, including qPCR versus digital PCR. And I know you're thinking about this quite a lot. Jill, do you want to take a crack at that one just initially?
Yeah, I would be happy to. And I'm, yeah, with the 5-minute flash talks, I think it is the methods that we end up kind of missing, which is a shame. Not a shame, but I'm glad that we're talking about this in the panel format. And it's going to be really important, both the methods. I know a lot of groups are doing qPCR and being very successful at that. We're doing ddPCR. What I just showed you was ddPCR. And that has been working well for us. And we do have plans for a comparison between that. I think other groups are having data that come out with that as well. The controls are extremely important too, by far. So for us, we're doing BCO for the recovery control, so bovine coronavirus. It's widely available and very cheap, and you can get in large quantities for not very much money. I know some groups are having some issues with that, as the recovery tends to be a little bit low, but we're using that, and you know, there's questions around normalization with that, and there's a whole other question around normalization and what to test with that. We plan to do the PMMOV. We're also doing an extraction recovery with Hep G, an armored version of Hep G, and then of course positive controls, negative controls, and so that's what we're doing in our lab. But I do think this is a really important point.
Yeah. Thanks very much. Kyle, let me go to you. Pepper mild model virus and normalization generally. Comments, thoughts?
Yeah, I think there's a clear need for normalization. So essentially the goal would be, right now, if we look per volume, sample volume, for example, to normalize to instead per fecal input, maybe for lack of a better way to frame it. And pepper mild model virus, for those that aren't familiar, is a plant virus that comes from food and appears to be relatively constant within a sewage system, so related to fecal input, so it passes through our gut. Another advantage about that virus is that it has an RNA genome, so it helps to correlate with some of the problems that you might have processing the SARS-CoV-2 RNA as well. So us and other groups have been using this as a normalization factor, but I do think there's some fundamental questions about whether or not that's really appropriate. So we see much greater variability temporally with SARS-CoV-2, for example, than PMMOV. And so are we really normalizing what we think we're normalizing? I hope so. But yes, I think there's a clear need for a normalization factor. And one of the leading candidates right now seems to be PMMOV.
And by the way, other people on the panel, raise your hand if you have something to contribute, and I'll call on you. Raul, if you wouldn't mind talking a bit about inhibition, your approach to that, and also quality controls, other controls you use experimentally, and maybe detection limits.
Sure.
That's a lot, but we had a couple of questions on that. So,
well, big picture, HRSD has moved to total process control. So we were monitoring for every sample. We were monitoring Hep G for every sample that we spiked in during extraction, but we've done a switch recently to just spiking in with bovine coronavirus in every sample and just taking total recovery. With respect to LODs, I personally think digital PCR is by far the more appropriate method to use for wastewater. I feel like it's more sensitive at the lower levels, which now, as we're moving from as we're moving from trends to kind of screening, I feel like you want to be a little as sensitive as possible, and I feel like digital PCR addresses that. One thing that I feel like isn't talked about is optimization of these assays to the digital platform. I feel like a lot of these COVID assays were designed for qPCR and on those specs for those instruments, whereas the instrument that we used, the QX2 droplet digital PCR, has like a tighter range of preferred assay limits. And so I feel like we could get more sensitive in wastewater, especially with digital PCR, if we maybe just like shifted the assays a little bit and redesigned them to be more appropriate for digital. That's actually something that we're working on right now, as well as a redesign of the peppermint model virus assay. I think that was also designed for qPCR and has some kind of design issues that I think a lot of people aren't addressing prior to using the assay.
Yep. Okay, we had a couple of questions about pasteurization and freezing samples at -80. And these are questions that many of us have talked about a good bit. Does anybody want to raise their hand and say something about that? Just, I guess, best practice in terms of, storing, processing samples.
Sure, I could kick it off for what we've seen. So one, we've seen that we have really good data now that pasteurization is effective in terms of inactivating any viable virus that might be there. We have seen with digital droplet that it decreases the concentration, or appears to. So the apparent increase in concentration may be platform specific due to maybe inhibition or something else, or maybe it's matrix specific. I believe it was, it might have been Hannah that presented that data. And the increase in concentration appeared to vary based on the facility. So it might be facility to facility. As far as freezing, we see a fairly significant decrease in apparent concentration if you freeze the sample prior to processing. However, we don't see that same decrease if you process the sample. And we see that the signal is relatively stable at storage at 4C for overnight, for example. And so the best practice might be rather just to process it and then freeze and keep the sample cold until then.
Kyle, by process, do you mean, does that include extraction or just doing your filtrations and into the freezer?
Yeah, so I just didn't want to take up too much time getting into the details, but yeah, we're doing a filtration and then we can freeze on the filter and it doesn't appear to have the same losses as the freezing of the bulk liquid. And that may or may not be the same. Yeah, correct.
Okay, thank you.
Another question about heterogeneity of matrices. So, if we go up, farther up into the sewer shed, that maybe looks pretty different than what you would get at a wastewater treatment plant. And I'm personally really interested in other kinds of fecal waste streams, like, somebody mentioned septic tanks or fecal sludges, those kinds of things. So comments from the panel on issues that raises methodologically in terms of sampling or processing of samples, and what the implications might be more broadly for wastewater-based epidemiology. And please just unmute yourself and speak.
Yes, so one, I would go first because of course, what we have seen that the a treatment plant with less capacity shows more variability. And also that because in a city, if we try to convince a municipal corporation or government, let's give out some fund to surveillance, they ask how many samples are enough. And like I have seen that composite sample of 14 water sets that was showing but still, what is the thumb rule? And so those are the critical issues. Here, general conception was that the dilution will occur once the gene will come into the treatment in the wastewater. But sometimes it is adding to numbers because the different pipelines are bringing more RNA. So they are so that dilution factor is also not. And like Kyle has also told that this coefficient that can convert gene number into the number of infected people is also little bit a challenge for all of us to find.
Also speaking to variability going up in the sewer shed, I know for a lot of our facility scale testing, because of this variability, we have responded to that by increasing sample frequency. A lot of us have settled into this weekly sampling regimen at the treatment facilities, and we are trying to switch to at least like 3 times a week, maybe even daily sampling at these upstream locations.
Great. Another question, maybe if Pungbo and Christine and Andre could comment we had a couple of questions on how more swabs compare with, say, grab samples, and have you done any comparisons across those different types of sampling strategies, and do you have any observations about relative sensitivity? I mean, I think we all think that maybe more swabs can yield a more sensitive approach, but do you have any observations to share on that?
I can start with that. And I did put that in the Pathable chat. So our basis for comparing is from the hospital sewage samples. So we've been collecting hospital sewage samples every week, both a grab sample and a more swab sample. And the more swabs are in there for 24 to 48 hours. And what Pungbo showed in one of the tables was that we're consistently getting more positive samples from the more swabs than from the grab samples. So that's our basis for thinking that the more swabs are more sensitive because they're staying in longer. And that's why we decided to use them for the dorms, because the wastewater flow is very intermittent in the dorms and we wanted to collect a positive sample. I'll turn it back to you and see if somebody else wants to comment on that.
Yeah, basically what we have been doing is a comparison between 24 hours composite samples and 24 hours Moore swabs in a wastewater treatment plant. We already knew that Moore swabs were useful to detect the virus on rivers and manholes, but we didn't know how they were like, if they will be useful on a wastewater treatment plant. So we have been putting the swabs the 24 hours and eluting them. And basically what we have seen is that CQ values are lower than the CQ values from the 24-hour composite samples. And I mean, we have reached like CQ values of 27 or 28 with the swabs, while the composite samples reach values of 36, 35. So we have also information about flow. So we are analyzing the flow rate and that kind of info in order to get more like conclusion about their useful. Yep.
Also, I want to comment like 2 weeks ago. So we had a positive swab sample from one dormitory on Emory campus. So we got a very low CT value, like 23, 24. So for grab sample, we never got, low CT value.
Yeah, and it is also challenging because here we started to see a decrease in our concentrations when rainy season start. So we thought about Moore swabs because they were basically staying in the stream for all the time, and we were able to catch that peaks of virus. Yeah.
I would add from our experience, I didn't present our data, but we're monitoring across the Georgia Tech campus where the goal is to monitor from 60 buildings. And a lot of them are highly intermittent flows, and it's very heterogeneous. And thankfully, we've been working with Pungbo and Christine on more swabs, and we've certainly found them to be more sensitive than grab samples.
Yeah, and when we obtained that CQ values just using 200 microliters of L-weight and doing an automatic extraction of RNA. It was pretty simple and pretty efficient. Yeah.
So we could spend a lot of time on methods, and obviously hopefully some of the attendees have gotten a flavor of some of the methodological issues that we're all working through. I want to spend the last few minutes, if we could, talking about where this is going in a broad public health a sense. So maybe I should go to you, Amy Kirby, first, if you wouldn't mind. How do we envision public health agencies using this data? Are there any public health agencies that are using the data that are being produced in wastewater sampling? And also, how could those needs change over time? How could we as a community be responsive to that? And by over time, I mean, obviously we've seen peaks and valleys, and now we're seeing another peak. I mean, ideally, at some point, a vaccine will arrive. that may change the picture, to a great degree as well. So I'm interested in your thoughts, Amy, and then after you're done, other thoughts from the panelists.
Sure. So just to quickly address sort of how we've seen this being used already. Of course, I think everybody now is familiar with the university applications where there's been testing of dorms that then is used to trigger clinical testing of the residents in those dorms to identify cases and move them into isolation. So that's a very effective use that we've seen now happen at multiple universities. But I think the bigger question is, well, what are the actions that happen from community level surveillance where you're testing at a wastewater treatment plant or out in the wastewater system? And we've seen a couple of places where public health departments have told us about specific things that they have valued of that. One is a system that was looking at sub sewer shed testing. And so they were detecting very high concentrations in only one area of the sewer shed, and they used that information to make decisions about where to site mobile testing units. So they specifically built up their testing capacity where wastewater testing was telling them they had a problem coming up. So a very discrete action. On the flip side, we have another health department that has put a lot of effort into wastewater surveillance. And when I put this question to them of, is there an example of how you have used this? They said no, but they continue to use it because it gives them great confidence that they understand what's going on at the community level. Unfortunately, the situation we're in is that clinical surveillance is very noisy. It's impacted by a lot of different factors, some of them under our control, some of them not. And so you're always on an epi side, sort of not quite sure that your clinical surveillance is telling you really what's going on in that moment. But sewage surveillance is different from that. It's not impacted by those same factors. And so if it's aligning with your clinical surveillance, it gives you great confidence that you can act on it and it's right. If it's telling you something different, you need to interrogate that more to understand, which system is telling you the truth. And that confidence is really, really valuable. And I think the more we see this happening at scale with communities, the more we'll appreciate that piece of it, that it's a unique data set that can help us understand what's truly going on at the community level.
Floor is open, other panelists.
Actually, just my 2 cents to it is that Kyle, Jill, everybody, many of us has told that this has been a noisy data, and many times my personal experience has been that I knew that now condition is going to deteriorate, but I was thinking whether I should announce it or not. But I saw that this confirmed cases has followed it up. So after 2-3 months, I feel that it is a better confidence. Even if you think that, okay, everything is normal, but your wastewater is showing something else, you can go about it. And so what I have found is that the change, irrespective of everything making constant, the change is important. If we are precise in analysis, the change is meaningful and it can be used.
The trends, yeah. So unfortunately, I think we're out of time for this session. I could spend all day talking to all of you about all these issues, and I really appreciate, again, thanks to everybody on the panel for your input and also sticking to time. I didn't have to be unkind with anybody today online, which is nice. Just one final request to all the speakers. We had a few people ask whether the slides that we all went through very quickly could be made available. And I know there's some sensitivities around data and whether the data are published or not, but I know that people would be very appreciative if you were able to post anything that you can post from your groups in the files section of the site for the session. And that way it'll be available to people. And of course, this recording will also be available as a resource for folks. So with that, thanks very much. This has been, I think, really, really interesting. Only scratching the surface, but super interesting. So thank you, thank you, thank you. And we'll see you later.
Thanks for organizing, Joe.
Yeah, thank you.